So let's begin our review of the 2024 AP Biology free response questions. As I always say, this work is not endorsed by the College Board and represents my own personal interpretations of these problems, which means I may get some wrong, okay? Or I may have solutions that are probably just maybe a little bit different than what the College Board will eventually post. So we can get started, but I also will say that my interpretation, or the way that I like to describe these problems, is always based upon trying to teach students the answers as opposed to just giving the answers. So let's be crazy and start in order. Okay, so this first of 6 questions here. All right, and as we scroll through our life here, we start with number 1. I love this question, crossing over in meiosis 1. And so, as I always tell my students, don't get all crazy about these questions, because most often some of these have, I don't know, very little to do with what's given to you, and sometimes they're just, you know, in the question. So in any case, number 1, in the new format here, they have described the function of the S phase of interphase. So really just a knowledge question that has, although it has something to do with this question. So in any case, my answer here, as most people would come to this conclusion, I described the function of S phase is accurately to complete DNA replication. Okay, so many ways to say that. So moving on, this life here. So back to the question. So this is a study about crossing over, and to see if the proteins that are normally in the centromere region of the sister chromatins here, okay, so the idea is that possibly proteins that are in the centromere region prevent crossover. And we have the hotspots, of course, where there is a lot of crossover action, and they've made in chromosome 8 of, I believe this is a organism of chromosome 8, they actually made a gene encoding red fluorescent protein allele and a green fluorescent protein allele in 2 different locations. So these are genetically engineered, okay, in a typical tetrad, or 2 homologous pairs that would show up in prophase of meiosis 1. So understanding those kind of things here. So this is the tetrad, as you've probably been taught, and they usually are connected, and the crossover, of course, is going to occur between the inside of those 2. Any case, so there's before crossover and there's after crossover, and you can see very clearly that there was an exchange of DNA matter here from the one inside sister chromatin pair to another. Okay, any case, so moving on to the question, explain why some haploid cells formed after meiosis in this will only have 1 fluorescent marker. Okay, notice they said haploid cells. They're talking about the gametes that are made here. All right, and will some, and the idea is here's some. So why will some? Well, the idea very simply is if we have this tetrad after crossover, okay, we know that in meiosis 1 we're going to split, okay, these 2 homologous pairs, and in meiosis 2 we're going to split these. So in the 4 daughter cells that are produced, very clearly we're going to have 1 of each. So in the 4 daughter cells, you're going to possibly have 1 that has neither, or 1 that has just the red, 1 that has the green, just the green, or 1 that has both. Okay, basically they're asking how meiosis works. So word for word, what did I say? Okay, well, I said that some haploid cells only have 1 fluorescent marker because crossover occurs between 2 adjacent homologous pairs in the tetrad. Each haploid cell result in only 1 of the 4 chromosomes in each daughter cell, as meiosis will separate both homologous pairs in meiosis 1. and cystochromatins in meiosis II. I'm very wordy here, just trying to help those trying to understand this. Since 2 chromosomes of chromosome 8 in the tetrad have one fluorescent marker, there's a 50% chance that each haploid cell produced through meiosis will contain a chromosome of 1 fluorescent. I definitely overstated what was necessary here. If you're taking this as a test, you should definitely get in and get out as soon as possible. Try to limit the words. Again, I'm trying to teach here. Okay, and so the next question was identify the control group. Okay, so let's talk about that question before we go to that answer. Okay, so B says identify the control group in the scientist's first experiment shown in figure 2. So in this part of this next piece, okay, what we have are different proteins that are placed in the hotspot region, and they measured the frequency of crossover with the idea that, hey, some of these proteins may influence the crossover in this hotspot region. And we can see that with the CTF protein, there was a decrease significantly because of the overlapping or the non-overlapping of the error bars. And there was from the control, there was no difference. Okay, so what was the control? Well, the control was, and I think they said it here, okay, in the first, they had 3 groups. I guess it was yeast cells. Okay, this is chromosome 8 of yeast. And this is the chromosome 8 they looked at. So yeah, so what I have here in the marker, group 1 contains no kinetochore proteins. That is your control. Okay, so any case, they said, I gotta go to the question, you have to go back and forth, sorry. Identify the control group for the scientists, and that's, of course, where there's no modifications to chromosome 8. All right, so that was 1, and so I just said very simply, if you had problems with this, the control group had no kinetochore proteins attached. That's something you should definitely have gotten there. All right, and that sometimes is called a negative control. We expect nothing to happen there. Okay. All right. So 1BII, in the follow-up experiment, the scientists created a modified version of CTF. And if you remember, CTF was the protein that had a significant, okay, change, right? It decreased the crossover. Therefore, we expect that that protein, okay, did influence and lower the amount of crossover as they were trying to test. So in the follow-up experiment, they modified a version of CTF in which the DNA binding portion had been removed. Remember that these kinetochore proteins connect to the centromere region, and they connect using part of the DNA strand. So what they're saying here is that we modified this kinetochore protein that no longer can bind, okay, with the DNA or the chromosome. They compared the frequency of crossing over in yeast cells in the presence, I'm reading from the question, so in the follow-up experiment, the scientists created a modified version of CTF in which the DNA binding portion had been removed. They compared the frequency of crossover in yeast cells and the presence and absence of unmodified CTF with that in yeast cells in the presence and absence of the modified. Okay, they didn't show you the data with that. In the follow-up experiment, justify why the scientists used a modified CTF protein that was unable to bind. So why did they use that? So again, in the follow-up, they wanted to test something else. Okay, and why the scientists used the modified CTF that was unable to bind as a new control. Well, the idea is very, very simple. We are testing, if you go back to the paragraph here, the little blurb above here, they're investigating whether the attaching individual kinetochore protein to a specific DNA sequence, okay, will affect the frequency of this crossing over. Well, we are assuming that it has to attach to the DNA or chromosome strand to prevent the crossover, but what we're not testing is maybe that protein has some other mechanism that stops crossover, as you know, as opposed to a physical blockage of crossover. So what this new control is, by not having this protein connecting with the DNA but still being in the same area, maybe this protein has some other mechanism that influence crossover. So we're assuming that, so this really tests or eliminates the other mechanism, right? So we're seeing if that is it. So what I said, okay, and again, we're going to scroll our hearts away here, back and forth between the questions. So I said, the scientists use a modified CTF as a control to eliminate the variable of whether the kinetic ore binding to the DNA plays a role in affecting crossover frequency. The presence of the kinetic ore itself without it binding to the DNA hotspot region may be the variable that influences crossover by some other mechanism. So remember, controls are always trying to eliminate the other variables. That's really important when you think about, and they love to ask about controls. So we identify a control, and now they're really asking, what does a control do? We're eliminating another variable. And I said that variable is whether kinetic ore binding plays a role in affecting crossover frequency. Okay, so maybe it does not do that. Maybe the kinetic core does something else. So by providing some information or data by the kinetic core not binding, maybe we can see that there's another factor, okay, involved here. So we're basically testing if there is another way, another variable in this experiment that we're controlling for, and that's how I said it. Certainly interested to see what the AP comes with, but always think as controls eliminate other variables. So if they just told you it was a control, so this is eliminating another possibility. So adding this control, we're eliminating the idea that the kinetic core, we're assuming it's not going to have an effect because it's not going to bind, but we don't know. So we're just going to eliminate the idea that it can do something other, maybe has some other influence. Okay, and that would change, of course, the entire data collection and understanding of the experiment if it did, right? So we're kind of proving that its mechanism is blocking, right? All right, any case. So onto BII, Captain. All right, so any case, let's go there. So BII says identify the independent variable in the follow experiment. So, so again, very simple, straightforward question. The independent variable, of course, is always going to be the variable that we manipulate. So in the follow experiment, it would be the experiment where we manipulate the CTF protein. Dependent variable, of course, is the crossover. How much crossover applies. So the independent variable would be the manipulated CTF. Always think of the independent variable as something that we manipulate, and we can manipulate any way we want to. In this case, we're taking away its DNA binding ability and seeing what that does to the dependent variable, how the dependent variable depends upon that, which is, again, that part's not changing. So any case, it's the modified CTF. Okay, so on to C. I don't think we need to see that. Based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome 8 hotspot. Okay, so here is a question where they just want you to evaluate the data. Okay, so based on figure 2, okay, what they're saying is, again, they use the word describe. So you're just going to tell me something about, you're not explaining, you're just going to basically describe. Going back to BII, these key words here, identify. Okay, just tell me. I love, that you should see identify. Just tell me. Okay, identify. Just tell me. Okay, just like in BI, they said identify the control. Of course, it's this one right here. So there's no explanation. All right, justify requires certainly some sentences here, but here we're describing. So you're telling me something about the characteristic in figure 2, basically reading the graph. So based on the graph, tell me something what the graph is telling me about the effect on the frequency of crossing over when CTFT is attached to chromosome 8 hotspot compared to the effect when IML. Okay, so again, the key here, all right, would be looking at the data, but looking for the significance. All right. So again, my short-term memory is gone. I'm very tall, so at my height it's gone. It's a joke. So let me just read this again. Describe the effect on the frequency of crossing over when CTF is attached to 8 compared to with IML. All right, so we're comparing. All right, so the CTF, okay, compared to IML. Well, there is a significant, okay, we're going to compare it, of course, to the control. That's important you understand. We're always comparing our data to the control. So CTF to the control here, there's a significant decrease in the frequency of crossing over with the CTF. And there is a statistically no difference in IML when compared to the control. Okay, we never compare between test groups. Okay, these are different modifications. This is no modification. So that's what we're going to say here. And the key is significance, right? There is what? No overlapping between those error bars, but there's overlapping here. Okay, so again, if you want to see what I said here, okay, and I said the scientists use a modified as a control. We did that. Oh, I. So identify. Just tell me. Unmodified was the independent variable, and then C. Sorry, getting there. There was a significant decrease in the frequency of crossovers as compared to the control, while there was no significant change in crossover frequency of the IML. Remember, again, those words significant, no, and not significant. I should say not there. Okay, not no significant. Okay, that's it. Okay. Any case, that is, in my opinion, needs to be stated there because of the error bars, the standard error of the mean. Okay, that was there. It shows the reliability of the data, right? Okay. Any case, so moving back on. So we have now D. Predict the effect on the number of copies of chromosome 8 likely to be present in the resulting daughter cell when CFT is attached to the hotspot. This question, I said to myself, are they really asking this? Because if you don't understand meiosis, then this becomes a very difficult question. But predict the effect on the number of copies of chromosome likely to be present in the resulting daughter cells. Well, the daughter cells are going to be the haploid cells, and as we stated before, because of the double halving that occurs, each daughter cell is only going to have one of the 4 chromosomes in this tetrad. Okay, remember this study is about crossing over, not about segregating DNA, right? So that's really important. So I find it to be an interesting question because there should be no difference. There should be no effect on the number of copies of chromosome 8 because crossing over has really nothing to do with the segregation of these chromosomes in anaphase in meiosis 1 and then anaphase in meiosis 2. So this should have no effect. Okay, remember crossing over isn't separating these chromosomes. Okay, so that's what I have there. Interesting question. DII, provide reasoning for justification. I think I just did. I think predicting basically is that there's no, very easy predict, there's going to be no effect on the number of copies likely to be present in the resulting, based upon CTF attached in the hotspot. No effect. And I just provide my reasoning because CTF doesn't prevent segregation. It affects the crossing over, but crossing over is not affecting the segregation of the movement of separating the the homologous chromosomes in anaphase 1, meiosis 1, and separating the adjoining sister chromatins in anaphase 2 of meiosis 2. So any case, I think that's what that is, and I can put that up there for your viewing displeasures. Okay, and so like DI, I said the binding of CTF and hotspot will have no effect. That's the prediction. Justify. In meiosis, tetrad will be halved twice, and in meiosis 2, regardless of a crossover. This results in 1 chromosome in each daughter cell. So crossover or not. I probably said it better in explaining to you now that I see this now. So crossover, I probably should say justify crossover. The amount of frequency of crossover has no effect in segregation. Really should say that now that I'm looking at that. Okay, so DII, explain. I'm not sure that explain might be a different question, so let's get on that. So last little piece here. Let me zoom in here. Last little piece. Explain how the presence of hotspots could increase the likelihood that the population will survive in the presence of selected pressures. So this is your idea that if you have a lot of places where there's crossover, that really will mean sexual reproduction. You're going to have a lot of exchanging of alleles to different chromosomes, and the shuffling of genes to different chromosomes adds to the diversity of the offspring in sexual reproduction by a whole heck of a lot. So this is going to create what? More diversity. And if you have more diversity in your offspring, you have different combinations of alleles or genes that give you different alleles. And this different number of alleles in your population is better able to what? Take on a new selective pressure as the environment changes and evolution or natural selection can occur. So that's what that question is all about. And of course, putting into words, what did I say? I don't know if I said it as eloquently, if I said it eloquently at all, is that the presence of crossover hotspots demonstrates a greater rate of crossover events that are occurring, resulting in an increase in diversity of the offspring. An increase in genetic diversity results in a greater frequency of available alleles in the population that could be selected for a future change in the environment, resulting in increased fitness. Same idea. Okay, so on to question 2. So number 2 says to investigate how increases in environmental temperatures affect the metabolism of certain organisms, researchers incubated liver cells from toads at different temperatures and measured 2 markers of metabolic activity. Those, of course, are rate of combustion, rate of oxygen consumption, which is really the same thing, and the rate of ATP synthesis. 3 different test groups, actually 6 different test groups at 3 different temperatures, right? So 2A, classic AP biology question. Okay, even though there's a background and there's some study involved, they just want you to know some knowledge. Describe, okay, tell me something, some characteristic of what water does in the hydrolysis of ATP. Okay, so we should recognize that hydrolysis is really the opposite of the dehydration synthesis. The word hydrolysis means splitting the water, and a hydroxide end and an H plus end are going to go to the 2 different ends. So many ways to describe this, probably very simple answer that they're looking for. I said water, and probably again, I'm always using more words than I probably need. Water is used to break the covalent bond, and that's important. These are covalent bonds, as you're going to deal with in all of organic chemistry, between a single phosphate group from the ATP molecule. Okay, again, that's enough right there to answer the question. I said the water splits, and I probably could say the water splits right here to break the covalent bond between them, and that'd probably be enough. But I definitely wanted to say that water splits, which is the same thing as hydrolyze, into an H plus and a hydroxide that creates a more stable product than what I had. And these are products because we're going to break ATP into ATP in a free phosphate ion. And the physical science guy of me wants to say that we're going to create something with stronger or more stable bonds. And you should know from your biology that this, and again, I'm getting more detail here than you probably have to need, just basically say that water splits to break the bond to break the bond to get the ATP and phosphate. You don't have to even say that. So I'm sure that water gets split to break the bond is really what you probably need here. But you just understand that breaking bonds here is a catabolic process, and most catabolic processes we get energy from, free energy from, okay, and we use that free energy to couple with something that requires energy. In this case, making ATP when we break down food to get energy. Any case, more details we need there, but again, I'm doing this to teach you. So back to 2B. All right. So the question states, using the template and the space provided in your response, construct a bar graph that represents the data shown. Notice they're saying it should be appropriately plotted and labeled. Okay. And there's going to be at least 2 points in this process. When they released the exam, I did not have the ability or did not know what the actual plotting looked like, at least a graph, empty graph, so I made my own. Obviously, if I haven't said so before, you can take a close look at my work. There'll be a link in the description to view that. Any case, here it is, and I basically plotted. Notice there's 6 test groups. You have the oxygen and the ATP synthesis, consumption and synthesis here with their respective error bars. Notice I am always going to put a title to my, even though they're not giving you a point for that, you should get in the habit. I'm also going to let you know what the values are of each of these X and Y axes. And notice appropriately that we have the dependent variable, okay, being measured, okay, which is in this case the rate on the y-axis. And of course I have a key here, a legend, to let everyone know what values we're dealing with. So pretty straightforward. Carefully make your plots. I'm not a big fan of this question. I think it takes a lot of time, and I think most people know how to graph. Okay, any case, more or less, no one's making hand graphs in research anymore. So again, whatever. So this is at least, I think, 2 points here for correctly plotting and then having the right scales and values here. All right, so on to BII. Based on the data now, they want you to ask a question. Determine, that's the important word. So you're getting, what do you get from the graph? Okay, so get me the value, basically determine, get me the value in the temperature at which the rate of oxygen consumption is different, okay, from the rate of consumption. So based on, give me basically the temperature at which the rate of oxygen is different. Now, they say different. They really should say significantly different, but they didn't want to give away the fact that we're really talking about a standard error of the mean here, our reliability factor, which gives us confidence about our values, right? So they want to know which one's different, okay? So the rate of oxygen consumption. So if you look at your graph, the rate of oxygen consumption, okay, is significantly greater at 30 degrees, okay? So if you look at 30, you can see that this value, even with this spread of unreliability, I guess, is this error bars don't overlap here. So we can see that from 25 to 30, it's greater at 30, okay? It's not any difference between 25 and 20. So there's no significant difference because these error bars overlap. Remember, we're talking about oxygen consumption here. So the only difference occurs where there's no overlapping of the error bars, okay, is from between 25 and 30. Here we can see that there's clearly a significant difference. Okay, and that's why I added the word significant, but they were just looking for value, determine. Remember, just get me the information. All right, so it's really important to know what they're asking for. Again, I tend to give more than necessary to understand the question, hopefully. All right, if you're using this to learn. All right, so in any case, so they wanted the temperature which the rate of oxygen is different from the rate of oxygen at 25. So if I didn't read that carefully, so from 25, it definitely increases to 30. There is no significance to 20, as I just mentioned, because of the error bars overlapping there. So BII, Captain. Okay, oh, CI now. Based on table 1, so now they're asking you to go back, not to the graph, or I guess you could use the graph, to describe the effect of the temperature on the rate of ATP synthesis from cells from toads. Okay, so how does temperature affect that? So based on the table, which I guess is the same as the graph, and you know, if you look at you can see that the rate of ATP synthesis looks like it's significant here and significant here. It looks like it steadily increases with temperature, even when you consider the error bars. So it looks like there's a significant there's a significant increase in temperature. As temperature increases, ATP synthesis increases. Again, you really, if they're going to give you error bars, you have to consider the overlapping here, and they don't overlap between one temperature to another. All right, so that's why I said here, okay, I said here that the rate of ATP rate of ATP synthesis significantly, again, I think it's important, and you need to say that in my book, increases from each test group of 20, 25, and 30. Thus, as temperature increases, the rate of ATP increases significantly. Again, I guess I could say that as well. I'm being overly wordy, as I normally am. Okay, so to CII, they wanted you to calculate something. So they just want the answer. Okay, so calculate the average amount of oxygen consumed in N moles for 10 milligrams of mitochondrial protein after 10 minutes at 25 degrees Celsius. So at 25 degrees Celsius, exactly how much oxygen is consumed. Remember, mole is a word you'll see in chemistry if you've already seen chemistry about how much. So we're using exactly, it's a value used to count, right? So it's a how much. This is for the time period, and this of course is per how much of the mitochondrial protein. Don't forget the mitochondrial protein is a catalyst. Okay, so think about this for a second. All right, mitochondria produces ATP by using oxygen, and there's essentially two parts. You can guess you could talk about the oxidation of the pyruvate, but essentially there's a Krebs cycle, an electron transport system, and those two combine for the greatest proportion amount of ATP produced, and therefore all the oxygen is consumed there. Okay, so any case, the key here is that we've got to go to our data and we go and find that in my calculation. Again, I put more information here than necessary, but they gave me at 25 degrees Celsius. Okay, and again, I'm sorry for scrolling up. My short-term memory is gone at my height or my age. So the amount of oxygen consumed at 25 degrees Celsius. So at 25 degrees Celsius, they gave us the 16.5. All right, now the units are important. That's that N mole, that per minute per milligram of catalyst. So remember, that's the unit given. And what does that mean? Where there's 16.5 N moles per 1 minute per 1 milligram. Now, some people are good with the math and some people don't quite get this. This is probably not a unit you've probably seen, but you can work this out. Okay, so the rate, okay, is 16.5 amount of O2 per 1 minute. That's the rate. Well, there's 10 minutes. So it could make sense to you that I'm going to times that by 10. Okay, and that gives me 165. But remember, it's per 1 milligram of mitochondrial proteins or enzymes, which are catalysts. So if you have 10 times that amount of mitochondrial enzymes, you're going to have 10 times the amount of rate, or in this case, use of oxygen. Okay. Now I have amount of O2 produced, and I think it's a little another error that I just looked at here. Okay, so it's the rate of oxygen consumption. So bad Grodski there. So this is the amount of O2 consumed. Okay, consumed there. Okay, per time. And so if we have a longer amount of time, and this is per 1 minute, we're going to times that by 10. And if we have 10 times the amount of enzymes present, it's going to be 10 times as much O2 consumed. So it's 6.5 times 10 times 10. Now, if you like units, like you will do in chemistry called dimensional analysis, okay, and this is the way I love to see you understand this. We have 16.5 N mole per 1 minute per 1 milligram. Per means divide by. So what does this equal? This statement equals this expression right here. And so when I times it by 10 minutes, the minutes cancel. Okay, and now I have N mole over 1 milligram. And then if you notice, if I times it by 10, you notice the milligrams cancel. And what am I left with? N moles. So if you use the unit that was given to you, this is the same thing as this. You can use dimensional analysis and set up your equation to solve for it. Or you can just understand what I did here. Okay, and it was only 1 point if you didn't get it. And again, you could have thought this out too. If you know how much O2 is, I guess I said produced, consumed in 1 minute, and know 10 minutes has transpired, so that would give us the 165. And it's produced per 1 gram of N grams, so if you have 10 times the amount, it'd be 10 times that. Either way, it's 1650. Okay, and again, it's probably not a calculation you've ever done, but it makes you think about units and something, and that's why your teacher hopefully has been doing a lot of different studies and understanding what these units mean. Okay, so on to 2D. I must predict something here. All right, so let's continue. A oligomyosin, okay, is a compound that can block the channel protein function of ATP synthase. Predict the effects of using this chemical on the protein gradient, okay, across the inner mitochondrial membrane. So yeah, you know a little bit about the idea that ATP synthase is, of course, that enzyme that produces ATP, and we should be aware that if we think about the structure, and I'll draw this kind of up here, we think about a mitochondria. It's got that cristae, that these projections in the inner membrane, and then you have another membrane. Remember, there's two membranes here, endosymbiotic theory, and so we have the inner membrane, okay, which would be right here, and then we have the intermembrane space, which is right here, okay, and we should know that the AP synthase sits, you know, in this region, just as the proteins of electron transport system. And we should be aware that when the pyruvate comes into the mitochondria, the Krebs cycles in this inner matrix, we should know that major reason for the energy carriers of these electrons is to pump the protons into this space. And these protons want to what? Jump back. And there's really only one way for them to jump back, is through the opening of the ATP synthase. Okay, so that's called the proton motor force, and that energy used from that gradient produces the ATP. I'm not teaching you this for the first time. This is something you should be remembering. So if we're going to block, okay, the channel of ATP synthase and not let me just zoom this up. So if we got the ATP, we got the ATP synthase. If we're going to block the H pluses from running from high concentration to low this way, well, you're going to build up the H pluses that get pumped into the intermembrane space. So what's going to happen to the protein gradient? Well, of course, it's going to increase. We're still pumping H pluses in by electron transport system using the high energy electrons from the high energy electron carriers, and that increases, but the H pluses can't move to a lower gradient, so the gradient increases. So how did I say that? Okay, many ways to do that. So I said predict, and again, it's kind of cool. I just say predict here. So you just got to tell me what you think is going to happen based upon understanding the biology of the mitochondria and the cell respiration in general. So this chemical will cause the protein gradient to increase across the inner mitochondrial membrane. Okay, so there'll be a greater difference between the intermembrane space and the matrix, and that's what the inner mitochondrial membrane separates. Okay, so that's how that works. So you had to know some information there. Okay, and a really good question, I think, there. All right, so moving on to D, we're now going to justify. I think I just justified it right there. So all we're going to say is that it's going to increase. So all they want to do is you're predicting, okay, that it would increase, and then I just essentially explained it, both of those words. So let's go see what I said, okay, or read what I just basically said. So I said oligomyosin will block protons from passively moving from a higher protein gradient in the intermembrane space through the inner membrane by blocking the channel protein function of ATP synthase. Electron transport system, which creates this gradient by pumping protons into the intermembrane space, will not be affected, as the chemical only blocks the channel of the ATP synthase, not the proton pumps. As the pumping of protons continues into the intermembrane space with a decrease of protons that can leave through the blocked ATP, the gradient increases. Again, I'm getting a lot wordy here. I probably can do this in a sentence or two. Okay, but again, I'm trying to teach those who don't understand the problem. All right, so I believe that is it for number 2. On to number 3. On to number 3 here. To investigate whether red blood cells of an animal lose the ability to take in glucose from their environment as they age, scientists collected red blood cells from guinea pigs that range from 1 day old to 7 months old. Okay, so they grabbed red blood cells from guinea pigs that ranged from 1 day old. Now, I really had trouble with this question because I really thought they left some things, I don't know, they're ambiguous here. So did they collect from 1-day old guinea pigs? Or is their red blood cells 1-day old and some of their red blood cells are 7 months old? I know that sounds silly probably to some reading this, but that's what really, I had trouble understanding the study here. Just not enough information for me anyway. They said the scientists incubated an equal number of red blood cells in separate cultures that contain 300 N molar solution of radioactive labeled glucose. So they incubated an equal number of red blood cells in separate cultures that contained, so I'm guessing they took these red blood cells from the and then they cultured them, making sure there was the same number. There's a leap there. So to me it's not absolutely clear here. The amount of radioactive labeled glucose present inside their blood cells of each was measured over time. Now they said each group. Now so we have groups, but what are the groups? Are the groups from 1 day to 7 months old? Do they have groups based upon the age of the red blood cells, or the groups based upon the age of the guinea pig that I pulled the red blood cells from? See, that to me, maybe I'm overthinking. So to me this is not a little bit too vague. But we have to deal with the problem. So in any case, let's see what we can do with it. A, describe a difference between a passive transport and active transport. I laugh because here's a knowledge question. Describe just tells me some characteristic, and they're telling me describe a difference between. They didn't say multiple, just describe a difference. And so we should know passive means that you don't have to put any energy to create a transport between a membrane, and an active, of course, tells you that you need to add energy. So the type of transport or shuttling of material through a semi-permeable membrane either requires energy, need ATP, or uses entropy, or I should say doesn't require energy, make things simpler. So very, very simple answer there. You should know that one, right? So moving on, again, they just wanted one difference. You should, my friends, make sure that you talk about both. Again, I'm over wordy, but I said passive transport does not require an input of energy, while active transport does require. That's the major difference. And they wanted what? One. So very simple. B, justify, why scientists use an equal number of blood cells in each culture as the control. So again, they love to ask about why are we using a control, and that's really what they're doing here in not so many words. What's the reason for us having a control? And you should get down. Controls are there to what? Eliminate other variables. We want to test one variable against and how it affects possibly the other. And of course, the one that is causing the change that we're trying to measure, that possibly is causing the change, is your independent variable, and the one that's changing because of it is your dependent variable. That should be clear. But so we're looking at just two things. We're trying to eliminate everything else. So why do we want an equal number? Well, we're trying to eliminate another factor that could affect our understanding how these two variables are related. So my answer to this type of question, is the control group utilize an equal number of red blood cells? And again, I know a lot of people will say you shouldn't rewrite the question, but I do so in this arena to help you understand what question I'm talking about. Because sometimes we lose track of what we're writing about. But in any case, the control group utilized an equal number of red blood cells as the test group in order to eliminate the variable. That's how I like to think about it. To eliminate the variable of more or less glucose capacity due to more or less red blood cells between the two test groups. If we're measuring total capacity that glucose is absorbed by red blood cells, and we're not measuring the average, then having a different number of red blood cells will cause that difference. We're not trying to make the link between more red blood cells equals more or a change in glucose. We're trying to measure, a different set of things here. So that's really important to understand that. So that's what I said. Now I added if the total amount of glucose capacity is to be measured. You can see my edits here. and not the average, then this control is necessary. All right, so we're just eliminating another variable that can affect glucose capacity. All right, and again, think of that when you ask for the reason for the control. They're kind of like, we've seen that question already. All right, they're just kind of changing it up in a different era. They're asking about a control. Eliminate another variable. So this is the second time we've seen that in this test. Okay, so C, glucose transporters are required for the facilitation of glucose into red blood cells. We should know that transporters are proteins. Okay, the scientists claim that the expression of the gene, that means from gene to protein, encoding these transporters decreases as the guinea pigs age. All right. If the scientist's claim is supported by experimental data, predict the effect of increased age on the amount of radioactive labeled glucose presented. So here we are predicting again. So we don't have to explain. If you notice, we're kind of doing that in the next part by the justification of what we're saying to C. So if we claim that the expression of the gene that actually produces these transporters that allow what? Sugar to enter red blood cells is decreased, we don't have the proteins to do it, okay, then we should predict that the amount of glucose absorbed decreases as they age. I mean, I don't know, to me that's nothing that you needed to know other than you needed to know what gene expression was. To me, this is basically right in the question. This question really perplexed me. I think it can't be this easy. So in any case, I thought about this longer than I had to. The amount of radioactive glucose present inside red blood cells will be lower in groups with older red blood cells, or I should say older guinea pigs. I guess when you're older, that's what's confused me. We're dealing with older, so I guess I should say with older red blood cells from older guinea pigs, and higher groups with younger red blood cells, or from red blood cells from younger, okay, guinea pigs. Yeah, I can see how I'm understanding the question better as I try to explain it more and more. Okay, but again, that's the problem I had with this question. Okay, I guess we're talking about the age of the guinea pigs that we pull blood from, which to me is a little bit misrepresented here, because we know that you're going to make red blood cells and replace old ones at different times. So an older guinea pig will have younger and newer, but it's not about that. It's about the older guinea pigs producing red blood cells that won't have transporters. Okay, it took me a bunch of times to understand, to completely get what they're talking about here. I think they could have been more clear here. All right, C kind of makes that more clear for me as I read this question a second time. Hey, that's just me. All right, so justify. Again, to me, it's reading comprehension here. So what did I put for the input? I said that a decreased expression for the gene of the glucose will result in less protein synthesized. They either catalyze chemical reactions indirectly, lead to the transport, or they make the proteins that are directly involved. So my answer here is I'm covering all bases, but when you have a decreased expression for the gene that occurs with older organisms, like let's say guinea pigs, we will have less expression. Less expression leads to less proteins. These proteins, remember, there's many jobs of proteins. Two most important ones, of course, is to catalyze the reactions that could indirectly lead to or directly lead to the transporters or the proteins themselves that are involved with the transport. So the idea that a decreased expression leads to less proteins, and the proteins are responsible for the glucose getting in to the red blood cells. All right, so I just kind of talked about either making the catalysts that catalyze the proteins, or the chemical reactions that lead to the proteins, or the proteins expressed directly. Okay, so that was my thinking in this question. But again, in a time-based scenario, I think they could have been more clear, in my opinion. I guess knowing that red blood cells are made continuously, okay, in the bone marrow of the guinea pigs, that's what was confusing me here. Okay, but the question should have cleared you up there. All right, moving on. Number 4. Okay, so in this question we have the classic invasive species question. This is the ecology question. All right, and I'm not even going to read the blurb since we've already had one question that pops up that doesn't require understanding what's going on in this blurb of information. Let's just go to it. 4A, describe the change in resilience of an ecosystem when there's a decrease in the number of species. I am so surprised to see a question that is just about word for word in last year's, 2023, that I think it is question 4 itself. So if you use last year's questions, it was definitely helpful. If you don't know what resilient means, it's to be able to bounce back, or the ability to resist, change, so to speak. So the resilience of an ecosystem, when there's a decrease in number of species. All right, well, less species, less diversity, less resilience. They just want you to describe the change, and we're going to describe the change as being decreasing. They didn't explain the change. And if they did, you should understand that if you have less species, then there's more stress, on organisms moving energy up through the trophic levels, and losing one of those two could actually stop the flow of energy and disrupt and kill and break down the ecosystem. Having more animals involved in that transfer of energy makes it more resilient when a few of them could die or possibly perish. There's others to fill those roles, and that's something you should understand. So that's there's going to be a decrease in resiliency. When there's a decrease, it can't bounce back. It gets affected more, and that's why we can't lose animals, and that's why we're so involved in the conservation of plants and animals, so they don't cause this ecosystem to break down. We don't think much about our ecosystems, but we should, because essentially when you think about it, our air comes from them, and that could be from the oceans or the amount of plants we have. So again, very important. So in any case, B, explain how the addition of the common wild oat affects the number of native bunch grasses that can be supported. So the wild oat in the reading is not the native species, but it's the species that is invasive. And what causes the problem in the reading is that this invasive species carries aphids. And these aphids carry viruses, and these viruses are detrimental to the native bunch grasses. So essentially, the wild oat introduces a virus that will kill the other bunch grasses, while the wild oat is not impacted. It has, some immunity to that virus. Because it's been exposed to it previously, through evolution, right? So in any case, what I said in 4B is, again, we're explaining the California grasslands ecosystem has a decreased capacity to support the number of native plants due to the wild oat grass introducing a virus via the aphid that is parthenogenic to the native species, but not to the invading oat grass. So many ways to say that. That's how I did it. But that, again, honestly, that question, is reading comprehension. It's right in there. I'm so surprised that that's what they're doing right now in AP Bio. A lot of reading comprehension. So this one was a knowledge question. This is reading comprehension. There seems to be a less stress as I go through this question, or stress on, knowing some really detailed understanding of biology and how it implicates a big picture, but take it where you can get it. C, researchers suggest adding ladybugs, predators of aphids, to the California grasslands. Predict the effects of adding ladybugs on the abundance of the native bunchgrass population. Well, if the ladybugs eat the aphids who carry the virus, there's less virus that is being exposed to the native bunchgrasses. So they just want you to predict. Didn't explain. They're asking you to kind of explain it or justify the next question. So what do you think is going to happen? Is it going to go up? Going to go down? Really, it's a 50-50 shot. So think about it. A is knowledge. B is reading his comprehension. C is a 50-50 shot. If you know nothing, then you'll get D wrong, but you still get 3 points. Oh my gosh. Any case, we predict. And the effect of adding ladybugs to the California grasslands will result in an increase in the abundance of the native bunch grasses. Because they are not going to be exposed to the what? To the virus as much. So they'll survive more. And of course, the reasoning, which is justify, ladybugs eat the aphids that the oat grass introduced into the ecosystem and thus decreases the exposure of the native grasses to the harmful virus. Okay, boy. And this justification is, again, in my opinion, from the question in C. So that should be a really good score for most people there. And if you didn't get it, I'm not making fun of you. Just show that this is a doable test. All right. So on to number 5. Sorry, going back in time. So number 5, research study mechanisms study mechanisms that enable or prevent speciation. Okay, so they're telling you that, but back to the first question. Describe a post-zygotic mechanism. So again, this is another knowledge question that's popping up. So this is something you had to know from your studies. They want a post, that's after making the zygote, mechanism that prevents gene flow and thus enables speciation. We should know speciation, okay, is when a population of a species, a subgroup of that population, loses the ability to reproduce with another subpopulation, and they break off. Okay, so there's what we call reproductive isolation. All right, so preventing gene flow means that they no longer can reproduce with each other, that reproductive isolation. So what is preventing something that becomes a zygote between two different species that doesn't enable them, that keeps them separate? Remember, the word enables speciation. So I wrote, again, this is a knowledge question, and there's a couple different examples, but the easiest one I remember is hybrid sterility is a post-zygotic mechanism that prevents gene flow and sustains speciation, okay, by producing offspring from two different species that cannot reproduce, right? Think about it. All of your offspring are sterile, okay? And in that way, they cannot produce, even though we do produce offspring, the offspring can't reproduce themselves, so therefore there isn't another generation. And these two species that did mate to make a zygote, okay, stay separated in terms of speciation, because there's an isolation between the species developing hybrids that could produce this mixture of individuals or a new species. Therefore, they stay separate there. Another example, hybrid inviability, which just means that the zygote, once it's been fertilized, the offspring doesn't live long enough. It's sickly in order for it to reproduce. So it's not able to live a full life and reproduce successfully. As low fitness. It has issues physically. And then, of course, there's hybrid breakdown, which is essentially very similar to that of hybrid inviability. This is usually where plants, where they don't grow as robust. They actually might make a second or third generation, but success is limited and eventually they die out. So those are the three. The one I always remember is the hybrid sterility. That's the easy one. Famous example is the horse and a donkey both making a mule. Donkeys and horses are different species, and of course the mule is the hybrid of them, but not a new species because they cannot reproduce themselves and carry on those genes. So back to number 5b. All right, got all this information here. So again, another research kind of blurb here, and they're studying codfish, or at least the descendants of types of codfish, and they're looking specifically at an allele that allows the codfish to what? Live well in colder temperatures, and that's because they have a antifreeze glycoprotein gene, which means a gene that codes for a chemical, a glycoprotein, that basically prevents the fluid inside the animal, mostly made of water, to freeze. Okay, so they reduce the freezing temperature of the fish, which means the fish can live in colder environments. At least that's the implication. The research compared genomic species in 9 cod species and 1 non-cod fish. They record the presence or absence of the elements of the functional gene. They include the promoter region, the actual gene sequences, and the untranslatable regions, okay, that are not part of the gene, that come after, okay. In any case, now let's go through the data here. You can see that, well, let's get back to the question, because the question is going to lead us to what we need to know from this. That's really the best way to go. Okay, so B, based on the data in figure 2, explain, okay, that is going to require a sentence or two, how changes in the genome enabled cods to survive and reproduce after a period of freezing between 10 and 15 million years ago. So 10 and 15 million years ago, my gosh, there was a new period of freezing temperatures. This was a new environmental change. And they want us to explain how the genome enabled cods to survive and reproduce after this time period. They're talking about natural selection here. Explain this to me. So for me, it's very simple. There's a new environmental change that creates a new selective pressure, which essentially means that the alleles that are already in the population through mutations and time and sexual reproduction, because there's always a great number of alleles provided in sexual reproduction, even though they may be in low quantities, they are now selected for. And their fitness increases, meaning they can reproduce more often than those that can't survive in the colder temperatures because they don't have that allele. And that gene proliferates and grows in frequency. That's basically natural selection. So the words that I used, and sometimes they accept the natural selection statement, but I kind of explained it out fully. So I said 10 to 15 million years ago, there was a period of sustained freezing temperatures. I said sustained to recognize that it's a new stable environment, okay, that represented a new environmental change. This new period of freezing temperatures provided a new selective pressure on the cods for the functional AG genes that were already present in some cods, but in low frequency. That's really important. You have to understand that these things were already there, okay? The environmental pressure doesn't create new alleles. It just promotes them in higher frequency, okay? The cods that had the functional AG gene had a greater reproductive success, and the antifreeze allele increased in subsequent generations. It's essentially explaining natural selection, all right? Any case, on to C. 5C says, using the template in the space provided for your response, place an X on the phylogenetic tree to represent the origin of the functional gene, the AG gene that produced the antifreeze allele. So I just kind of cut and pasted the value here. So my X, okay, is right at the common ancestor. That's where I thought it would go. And I looked very carefully because, number one, to me, the wording's kind of weird. They said, when would this gene appear? Remember, in natural selection, the gene has been around. It just hasn't been at higher frequency. So to me, when they say when the gene appeared, it's when it was a high enough frequency that most individuals in the population had it. Okay, so that's a kind of a, again, I'm playing, I'm cutting hairs here, but it's important to know the distinction in that definition. So to me, it has to occur during the selective pressure, the new selective, or the new environmental changes, and therefore new selective pressure in that time. Now, if I look carefully, okay, you can see that these individuals had the promoter for the gene, had the gene, okay, but as they said, L here means somehow they lost it, okay? So there was another event here. This could be losing the gene, could mean that there was a mutation that stopped the gene. There could be genetic drift that lost the allele, who knows, but these guys lost it, which means they had it, and it makes sense they had it because look, they had the promoter region for the gene. They had the gene itself. So to me, I needed the clade. The clade is the group of forks that come to one common ancestor. So to me, all these species, okay, had to have that upstream somewhere. And the only place it can have upstream to me is right at this area here. Okay, so that's where I put it. Now, some people might say, well, could it happen beforehand? I don't think you're going to have it before you're definitely not going to have it before the new environmental change that creates the new selective pressure. It's got to happen, and there has to be some time period. So it makes sense to me what would happen here. Could you have put it here? I think that's wrong, because I think that these individuals had it as well. But I think someone could have said they wanted an X. You could have said these two X's. I don't think that's the right answer. The best answer to me is right at the common ancestor, okay? Because these are all related to an ancestor that had that gene. These guys, these two species lost it, okay? So when they made a new, when there was another split, this new ancestor had a new species to occur. There was some change here, okay, that occurred during that. So to me, that's where the X belongs, not in front. Now, could you make a case that the X could be in front of the common ancestor? I think you could. I think your X could somewhere be in front of that a little bit, but not in front of that selective pressure. To me, there's going to be some time. It makes sense that the common ancestor, that's where there was these individuals that became, through another form of selective pressure or speciation, these guys became new species here, okay? Any case, so if we have to put 1X, it has to affect the single clade. To me, the only explanation is right there. All right, so any case, that's where they wanted you to just put an X there. And so on to D. Based on figure 2, explain how genetic differences among the species in the family of cod, that's the early family right here, that's a drawing right here, determine the habitats in which those, explain how the genetic differences among these species here determine habitats in which they can survive. Okay, well, to me, at some point, if you notice, the individuals that can survive at higher temperatures don't have the gene. Okay, so these creatures either evolved into ones that don't, or they evolved in the ones they had for a while, but they also evolved into ones that have the allele. Okay, so the genetic differences that family, now remember, again, if you think about classification, this is family, okay, and then there's genus, species, right? So this is the family, and these are all connected to that. So all of these individuals were really, all these different species came from that family right there. So these descendants here, why do they have different habitats that they live in? Because based upon the genetic differences of either having the allele, right, for the antifreeze, or not having. So where they live was essentially guided by the alleles that allow them to live there. Okay, so again, getting too wordy, but I said very simply, I think I said it best here, cods that had the gene for the functional AG gene have better fitness in colder habitats than those without the gene. And that's clearly demonstrated, okay, here. All right, whether you had the gene and lost it, okay, you survive better in warmer climates because you can't or don't have the fitness to survive in colder climates. All right, so that's pretty straightforward. Okay, and on to number 6. All right, so number 6, if you're running out of time, you should always go read the question first, because sometimes you have a knowledge question. We've been seeing that. So this one does not have that. If it says use the data in figure 1, identify the rate in ms or milliseconds per codon, all right, that was recorded with the greatest times for the GAC codon. So again, if you didn't have time, you just go to figure 1 and you're looking for the greatest number of times for the GAC codon. Okay, so this was a study and we're identifying, so we just want the value. This is all that they wanted, but I'm just explaining it. They want the greatest number of times and what that rate was. So how many times did we get that rate? It was the greatest amount of having that rate. That's what this is about. So this is a translation rate. Now translation, of course, is creating the messenger RNA code, we call it codon, into the protein. And so this codon actually into an amino acid that eventually becomes a protein. So this is time per codon, which is a weird rate, and it really is. So the time it takes to create for the messenger RNA in translation with its tRNA and the subunits coming together to make an amino acid. Okay, so how fast? And this is the time per codon. Okay, so this is just 150. Some people I know put down 20, right? They wanted the rate that was recorded with the greatest number of times. They wanted the rate, and the rate, of course, is your dependent variable. Independent was how many times it was recorded. Okay, any case. At least based on this graph. So in any case, moving on. So that's what this was about. This was about, and if you weren't sure, you go back and they measured how long the ribosome remains in stationary with each codon. They determined the average translational rates for all codons. Yeah. But in any case, this is about how fast it takes to take the messenger RNA's particular codon. Notice there's particular ones. Some are going to have different speeds and different graphs. Okay, so in any case, next question. Using the data from figure 1, graphs B and C, which is for different codons, which is for A, UU, and UGG codons, describe the variation in the translational rate. Okay, and you're comparing one to the other, so you need to describe both. So if you look at these two, okay, they're asking you to look at the data, and this is really straightforward to me in terms of the statistics you've done or should have done in understanding this. I would say, looking at this, that the rate or the mean, if this is not quite a bell curve, right? But we could probably say the mean is closest to 100. You could actually say the mode is 100, right? Mode is the greatest number, and that the mode is 100 for codon AAU, and the mode is the greatest number of recorded rates for UGG is 350. Okay, and if I'm not mistaken, they were asking for what? Just one? They said using data, describe the variation. Okay, describing it. So didn't say a single. So any case, you could say the mode. I could say that the mean was closest to 100 in AAU, while the mean was closest to 350. Okay. And you could say that the standard deviation or the spread of data was smaller for AUU, whereas the spread of data or the standard deviation was greater for UGG. So understanding some basics, statistics, and a normal distribution, I know it's not perfect, you can talk about those. So that's what I said. And I said the translation of rate of AUU has a mean closest to 100, while the translation of UGG has a mean closest to 350. You could definitely use mode. Maybe I should use mode there. The translation rate of AU has a much smaller standard deviation than that of the translation of UGG. Okay, just spread of data. So I'm describing it. I'm telling you what it is based upon the graph, not making any analysis there. And I did go both ways, and I would definitely, since I'm describing, those are two main characteristical differences there. Okay, so on to C. Scientists hypothesize that tRNA molecules that bind to UGG are available in lower abundance. That kind of makes sense. If you have less tRNAs available for a particular codon, okay, then therefore the speed in which you do that is going to change. Okay, so again, let's think about this. Scientists hypothesize that the tRNA molecules that bind to UGG are available in lower abundance. Okay, so support the scientist's claim. Okay, so if we look at UGG, they've got a lot more molecules of, well, amino acids being made. We're translating more of this code into an amino acid at a shorter time per codon. That's kind of tricky here. Most rates are something you're producing or missing per time. Here it's time per codon, so it's kind of tricky. So you got to understand that because the time to make them is shorter, okay, therefore these are going much faster. That's, again, you got to be careful. Even though this is a time per codon is a smaller value, these are a faster rate, even though their numbers are lower. I don't like what they did there, but you had to figure that time per codon. So if you're saying that tRNA molecules are available in lower abundance than tRNAs that bind, so the tRNAs that bind to UGG are available in lower abundance, okay, then tRNAs that bind to AAU. So these are in lower abundance than AAU. That kind of makes sense. On average, it takes more time to produce codons. These are going slower. These are going faster. So I know it's crazy. The numbers are smaller, but it's faster because they're measuring time per. So we would say that what's the support? So support the hypothesis that there's less available tRNAs, okay, here. Well, because the rates are higher. The MS per codon is higher. Okay, so what I said here was the hypothesis that tRNA that bind with UGG codons are available in lower is supported. So again, I should read that more clearly. This hypothesis that tRNA that bind to UGG codons are available in lower concentration is supported by the greater range of higher translation rates as compared. So I just put that in misnomer here. Rates are measured in time per codon. Thus, the greater this rate, the longer it took. Okay, so again, I'm saying that the UGG codons are probably in lower concentration. It supports this idea because there's a greater range of higher translational rates, which means it took longer to make these codons, which supports the idea there's less tRNA to deliver them in the subunit of tRNA in translation. So it does support that. It just gets a little bit tricky because they're using the idea of time per codon. Lower number is faster, okay? So careful with that. And then you have to explain something here. I think it's along the same. Oh no. Okay, so amino acids can be encoded by multiple codons. We should all know that. There's that that wobble, right? That last third codon can be different, but the first two really are what define the amino acid in the codon of the messenger RNA. So amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently in messenger RNA than others. Think about this. In many, certain codons for the same amino occur more frequently than do others. Based on the data provided, explain why the use of one codon, now we use data, so based on the data given to us, explain why the use of one codon over another for the same amino acid might result in increased levels of protein productions. Now think about this for a second, because they gave me this. I was thinking in terms of, you know, why we have multiple codons, but what I think solidified my understanding in this question was that because they said based on the data. So when I look at the data, I said, okay, we're comparing the translational speeds of different codons. And these are all coding for different amino acids. They didn't show you ones that code for the same, but we can assume, based on the statement, that if I had AUC, okay, if that was another same codon for the same amino acid, okay, that it would have a different value because of the statement they just said, okay. Certain codons the same occur more frequently in messenger RNA, okay. So think about that. Why would that? And there's a reason for that. There's an evolution reason, because the faster you can get that protein made, the better chance you are to adapt to your environment and do the things you need to do to survive. So explain why. So for me, it was the idea that the reason why we have certain codons that are more frequent is because they must have faster translational rates, and faster translational rates will lead to increased levels of protein production. I definitely have to think about that one. Okay, that got me thinking a little bit. Okay, and I know you don't always have that time. Okay, so I said, very simply, if a messenger RNA contains codons that are able to translate faster for the same primary amino acid sequence as another messenger RNA with different codons, then the level of proteins will increase. Okay, and so they're really asking whether or not the protein synthesis is more beneficial, but we do know that a faster rate of protein synthesis is definitely a helpful adaptation. We know that many proteins are made from the same messenger RNA, okay, at times. And in any case, we do know, I should really say that there's many messenger RNA made from the same DNA template, right? We have multiple, I should say that. Okay, so the reason why we do those things is to produce the proteins fast and furious. Okay, if the cell is gene is open for that, it's definitely more advantageous. In any case, it wasn't about that. It was the fact that again, if you have to reread the question, okay, based on the data, that's the key here. Explain why the use of one codon over another for the same amino acid might result in increased levels, because that one codon, okay, might translate faster, and faster translation rates will lead to an increased level of protein. So it's actually a simple question, okay, but I had to think about that for sure. All right, I hope this helped. And again, I'm going to go back and fix this a little bit. I can see some of my little errors here, and the link will be available in the description area for better look at my work. If it helped, thanks a lot, and take care.